This conference will now be recorded.
Hello students. So we are preparing for NEET UG exam of 2024. Now we are in a very important chapter number 10, cell cycle and cell division. So first what you should do is you should go through the NCERT textbook and you should get a clear idea about this chapter. And now we are discussing the important points, only the important points, and then we are moving to the previous year question paper so that you can uh, answer those questions. Okay, so here slightly confusing questions you may get. Here we are learning about the complete cell cycle and how a cell divides. Okay, what and all changes are happening during a cell cycle in each stage, what changes are there in a cell cycle, and how a cell is getting ready for the division stage. Okay, so here we have learned in chapter 10 NEET syllabus, showing here phases of cell cycle, then mitosis and the significance of mitosis, and the same way you need to learn about meiosis, stages of meiosis 1, meiosis 2, then difference between mitosis and meiosis. Okay, these many things you need to learn from this chapter. So we have already discussed about cell cycle. Then in that, the stages, one interphase is there and one mitotic phase or the division phase. This is the division phase which we have marked as M. Interphase, that's a long phase, and interphase consists of G1 or growth 1, gap phase 1, and the S phase and G2 phase. Okay, this G1 you can see the cell growth, and then in the S stage, that is the synthesis stage, that means DNA replication, DNA synthesis is taking place, and G2, that is the preparation for mitosis. Okay, and here in the mitosis, after all this, cell is getting ready for the division stage in all these stages. So after that, it is moving to the division stage of division, stage of cell division, that is mitotic stage. Okay, here first the division of nucleus or the karyokinesis is taking place, and next comes the cytokinesis. So here karyokinesis inside the nucleus division is taking place. So along with that, here you can see the chromosome that is also getting divided, and it is moving the synthesized or the replicated DNA, that is also getting divided, because the DNA where it is present in the nucleus. So you can see how it is getting divided between the newly forming cells. So here nucleus is getting separated and two nuclei are forming, and after that the cytoplasm is also getting divided. You can see here the cytoplasm that is also getting divided, so that new daughter cells are getting formed from the pre-existing cells. Okay, so all the cells are forming from the pre-existing cells. So how daughter nuclei or daughter cells are getting formed from a pre-existing cell. Okay, so now you got how we are connecting the previous chapters, and we need to learn about the cell. That's why I said first we need to learn about the cell organelles. Now the hypothesis or the ideas of different scientists, how we are connecting all the cells are arising from the pre-existing cells. Here you can see the exact way how a new daughter cell is getting formed. Okay, then in animal cell and plant cell, difference will be there, right? The formation of the daughter cell, that will be different. Here in animal cell you can see there is a cleavage furrow formation, and after that the cytoplasm is getting divided, so that two daughter cells are getting formed. And at the same time in a plant cell, what is happening here, a cell plate is getting formed. Okay, and that touches both the cell wall, and that's how two separate cells are getting formed. Okay, now we said about the cell division, right? So here cell division, how it is taking place, we have learned, right? So cell division means that is a very essential process or very important process, and during this cell division each parental cell that is getting divided and that give rise to two or more than two daughter cells. Okay, here you can see a single cell is getting divided and finally you can see it is making two daughter cells, right? So these two daughter cells are more than two. Okay, so this cell division occurs in three ways. So cell division is taking place that's in three ways. Which are the three types of cell division? First one is amitosis. Amitosis. So this is a direct cell division. Okay, so that means this is by splitting the nucleus. First splitting the nucleus and that is followed by the cytoplasm. Okay, here direct cell division. This is the direct cell division. Okay, so first splitting the nucleus and that is followed by the cytoplasm. So here it does not divide the nuclear material equitably. Okay, so that occurs. There is no, we cannot say that the nuclear material, everything is dividing in equal way. And this is not happening with the help of spindle fibers. Okay, there is no formation of spindle fibers. So this type of cell division that occurs in prokaryotes. Prokaryotes. In prokaryotes, you can see this amitosis. Okay, and another type of cell division that is mitosis. Okay, another type. The second type is mitosis. So in mitosis, this term mitosis, this was given by Fleming. Term was given by Fleming. Okay, so the term mitosis was given by Fleming and that is in 1882. Okay, so this mitosis is also called as equational division. Mitosis is also called as equational division. You can see here everything in equal way it is happening, right? So equational division, and in this parent cell is getting divided and that forms two identical daughter cells with identical genetic complement. Clear? So here equational division is taking place and after division two equal two daughter cells are getting formed. Two daughter cells are forming and these two daughter cells are with identical genetic complement. With identical genetic constituency. Okay. That will be similar. Both cells will be similar. Okay, there won't be any genetical change. So phenomenon of mitosis that is taking place in the somatic cells of animals and plants. So in the somatic cells of animals and plants, it is taking place and this is mainly working for the growth and repair of body cells. Growth and repair of body cells. Okay, so it mainly works for the growth and repair of body cells. Okay, and this is involved in the process of asexual reproduction. Okay, there is no change in the daughter cell's genetic constituency. Okay, so here that involves in the process of asexual reproduction. And what are all the significance of mitosis? See, genetic stability. Mitosis means the genetic content, it's not getting changed. Okay, the same genetic complement. It is moving in the same way. The genetic condition of the organism is not getting changed. So that maintains the genetic stability. Genetic stability is getting maintained, and this is, we said, it is helping for the growth and repair of body cells, or it is helping for the growth and development, then for repairing, healing, and regeneration of cells, for everything. Mitosis, their mitosis is taking place. Okay, so that's the importance or significance of mitosis. Okay, here you can see in mitosis what is happening. This is a cell which is in the interphase. Interphase means this is in the final stage. Here we have mentioned as G2, means it is moving for the next step is division. Okay, so next step, which one? Here in the interphase, the cell which is an interphase, you can see the centrosome with centrioles. Okay, and here you can see the nucleolus, this purple color dot that shows the nucleolus, and here inside the nucleus the genetic material that is in a thread shape, or it is loose. Okay, and covering the genetic material, you can see the nuclear envelope. Here we have marked the white color mark that shows the nuclear envelope, and here the cell, plasma membrane of the cell, you can see, and the chromosomes, which I said that is not in a condensed form, that is in the loose form. Okay, so in the G2 phase, the interphase, especially in the interphase, when it is moving to the which phase? Divisional stage. That cell in G2 stage or G2 phase, you can see in this condition. Okay, then it is moving to the prophase. Prophase. So here in the prophase, you can see the mitotic spindle formation. That spindle formation will start in the prophase. In the early mitotic spindle formation, you can see here, and inside you can see the the chromosome or the chromatin threads or the chromosomes will be in the condensed form with two sister chromatids. You can see here it is in a loose form. Here it is in a condensed form and with two sister chromatids. It will be in the prophase. Okay, and when it is going to the pro-metaphase means it's moving from the prophase and it is moving towards the metaphase, or it is reaching in the metaphase. So that condition, here you can see the spindle formation is almost complete, right? And the fragments of nuclear envelope will disappear, right? So here in the pro-metaphase, nuclear envelope will get disappeared completely, and you can see the sister chromatids with the kinetochore, and the spindles are attached to the kinetochore. You can see here. Okay, like this you can see the sister chromatids. And here, how the spindle will get attached? Here to this kinetochore, from the top side spindle will get attached, and to this side from the down spindle will get attached. Here you can see it will come to the center and it will get arranged. Which one? This chromosomes with that is having two sister chromatids. See, like this it will be the sister chromatids. You can see here the two sister chromatids, and the kinetochore to that spindle is getting attached, and that spindle, this spindle is pulling the sister chromatids, one sister chromatid towards one side and the other sister chromatid towards the other side. That's how the sister chromatids are getting separated. Clear? So here, here you can see in the pro-metaphase you will get questions like pro-metaphase. So here you can see the spindles are completely formed, and it is attached to the kinetochore of each chromatid or the each chromosome. So here you can see the fragments of nuclear envelope. Nuclear envelope will get disappeared completely at the end of this stage. Okay, Then from the pro-metaphase, the cell is moving to metaphase. Okay, so here you can see the complete spindle attachment and all the chromosomes are arranged in the center, or this is called as in the metaphase plate, or in the center it is arranged. Okay, and the spindles are attached from both the sides. Here you can see in this metaphase picture, right? Next step in the mitosis. Next step, we said the uh, spindles are attached to the kinetochore, right? So from both sides it will pull, and the sister chromatids are getting separated. Here you can see sister chromatids. One sister chromatid is moving towards the upside, and the another sister chromatid or the daughter chromosomes, they are moving towards downside, right? Here we have marked very clearly daughter chromosomes, that portions, both the sister chromatids are moving one towards the opposite poles. Okay, next, in the next step, you can see this is happening. The separation is happening with the help of the spindle fibers, and that is happening in the anaphase. Next, this is now chromosome separation over, right? So nuclear envelope, it will form again the chromosome, that will become like loose threads. Again, the nuclear envelope will form because two nuclei formed. That means karyokinesis over. That is in the telophase. Right. So in the telophase, karyokinesis or the nuclear separation will get over, and the cytoplasmic separation, the separation of cytoplasm, that is also taking place during which one? During the telophase. So cytokinesis is also telophase, and cytokinesis is also taking place here in animal cell. What will happen here? Cleavage furrow is forming, so nuclear envelope is forming that covers the nucleus, and inside the nucleus chromosome or the genetic material will be present, and here then the cytoplasmic separation is taking place. For that, cleavage furrow is forming. See, you can see here the furrow is forming from both the sides, and here it will come again inside, it will join here, and it forms a separate cells, two separate cells. Here nuclear envelope will form. Okay, karyokinesis over means that nuclear envelope will form towards or on that newly formed material, genetic material, to cover that genetic material, to separate nuclei will get formed in the telophase. Okay, you should remember in which stage this diagram. So everything, if you are trying to remember with the help of picture, means it will be easy for you to get the answer. Try to remember all these figures, and it's very easy. First in condensed, in non-condensed or uncondensed form, then it is getting condensed, then it is getting arranged in the middle portion, it's slowly coming to the middle portion, spindle is forming. So spindle formation prophase it is starting. So spindle slowly it is trying to move towards the chromosomes, the thickened or the condensed chromosomes. Then these chromosomes are completely getting arranged in the metaphase plate, then the spindle attachment is taking place, and then the sister chromatids are getting pulled towards the opposite poles. So chromosomes got separated. So around the new separated chromosomes, the nuclear envelope is forming to protect that chromosome inside the nucleus. Then karyokinesis is getting over there. Next step is the cytokinesis. Cytoplasm also will get separated. Then only to complete cell formation is taking place, right? So that way you should remember the picture. Okay, so this is the mitosis, stages of mitosis, right? And this is helping to maintain the genetic stability, right? Here, chromosome synthesis or the DNA synthesis is taking place, the genetic material synthesis is taking place, right? So it's becoming double. But here again, while getting separated, again the same number of chromosomes are getting restored after separation also, right? The doubled chromosome, again it is equally dividing, and again it is forming to new cells. So equal division is taking place, right? So that maintains the genetic stability, and that helps in the growth and development and in the healing and regeneration of cells and repairing and all these things. It is helping. Which one? Mitosis. Clear? Next type of cell division is meiosis. And here the meiosis term, the term meiosis was given by Farmer and Moore. Okay, and this in 1905. So this is also called as meiosis is also called as reduction division. Reductional division or reduction division. But here equational division, which is that mitosis, equally it is getting divided. Here reductional division. I'll say why it is called as reductional division while explaining the stages. Okay, so here you can see the same mitosis steps are repeating up to here, right? Here you can see the chromosome condenses and nuclear envelope that is getting or that is breaking down. Then here crossing over, this is another process. Crossing over is taking place. Crossing over, okay, the chromosomes will cross over the other chromosome. Then here it will form pairs, and again spindle formation is taking place. See, after crossing over, here you can see the exchange of material, that chromosome portions. Okay, so one blue color it is here, another half is red color. So that is the exchange. Here after crossing over, exchange of that portions happened. Okay, that may not be completely, some portions may get crossover. So many things are happening in this. So pairs of homologous chromosomes, they are moving towards the equator or to the middle portion. Their spindle is attached, and again the separation. In the same way, homologous chromosomes move towards the opposite poles of the cells, and spindle attachment for the separation. And here nuclear or the karyokinesis got over, then cytokinesis will take place or the cytoplasm divides, right? Then after these stages, this we call as prophase 1, metaphase 1, anaphase 1 and telophase 1. Which one? The first stage. Then each cell will move, this cell will move to the prophase 1 and this cell also will move to the prophase 1. Okay, again this will go through the same stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. In prophase 2, again spindle formation, new spindles are getting formed around the chromosome. So here the synthesis of genetic material happened and it got divided equally. Now each cell will be having single set of chromosome. Here in the prophase 1, new spindle formation is happening around the chromosome, and in metaphase, then it is moving to the metaphase, spindle attachment is taking place. Metasome, in the metaphase, two chromosomes are getting arranged at the equator. You can see from both cell, the same thing, same processes are taking place, right? After spindle attachment, in the anaphase, again the centromere is dividing and chromatids are moving towards both the sides or the opposite poles of the cell. Here you can see in the second cell also same processes are taking place, right? Next, the telophase. Again, here from one set of chromosomes, again that got divided, right? In the anaphase, that sister chromatids got separated. So here what is happening, a nuclear envelope is forming around the each set of chromosomes and cytoplasm is also getting divided. So already here the here synthesis, again genetic material is not getting doubled, or their synthesis of genetic material is not happening in the second time, right? Only one time the DNA synthesis is taking place and two cells are forming, and each cell is entering to next one more cycle of all these events, right? So what is happening there? Here in the second stage, in meiosis 1 and meiosis, this is meiosis 1, and each cell after meiosis it is entering to meiosis 2. So here there is no synthesis, only division is taking place. So again the whole set of chromosome is getting divided, and here you will get to here the chromosome number will be 2n. But after this division, you can see here from the single set of chromosome, both cells are getting formed. So here the division of that chromosome 2n is becoming half and half. Here 1n, here 1n. So cells are getting divided and chromosome number is also getting divided in this way. Here also, right? So same way cells are getting divided. So here reduction of chromosome number that is taking place. That's why this is called as reductional division. So the phenomenon of meiosis is taking place in the reproductive cells. In reproductive cells, this phenomenon of meiosis is taking place, and that helps in the formation of gametes and spores. Okay, this is helping for gamete formation. See, in the male gamete and female gamete, the chromosome after meiosis, when it is forming, chromosome number will be half, right? So these half chromosome, again in the male gamete n number of chromosome, female gamete n number of chromosome, both are joining together and it is forming the zygote, right? This zygote will be having n plus n 2n number of chromosome. Again the chromosome number is getting restored. As the same individual, it is getting restored, right? So that involves in the process of sexual reproduction. And here after again during reproduction or in the formation of zygote, that chromosome number is getting restored. Okay, what is the importance of this meiosis? Meiosis, that is maintaining the specific number of chromosomes in each organism, right? You think about here 2n number of chromosome. In gametes also 2n number of chromosome means male gamete will be having 2n and female gamete also will be having 2n. Both are joining means what will be the condition of that organism which is forming? In the zygote, 4n number of chromosomes will be there, double the number of chromosomes. So that individual will get changed, right? See, chromosome number is getting restored. That's why the same characters it is showing. If chromosome number is double, it will show different characters, right? There will be change in the body condition, in the characters or the uh, features it is showing. Everything will get changed. So here this meiosis, that is a reductional division, and that is maintaining specific number of chromosomes, that helps to maintain the specific number of chromosomes in each species. Okay, and here independent assortment, here comes the law, Mendel's law. Here independent assortment of chromosome is taking place. The chromosomes are independently getting assorted, and that leads to variation. That forms the diversity, that forms the species diversity and the formation of different species or different types of organisms, that is leading this independent assortment of chromosomes leading to the diversity, or that leads to variation. Okay, so here we have learned mitosis, meiosis and amitosis. Okay, so what is the importance of the cell division? Tell me, what is the importance of the cell division? Why the cell division should be there? See, cell division, first thing important, that is important for reproduction, right? Whether it is sexual reproduction or asexual reproduction, that is the first main importance of cell division. Then reproduction means that is showing the continuity of generation. Continuity of generation, and that maintains cell size maintenance. Then we said mitosis, that is helping for repairing of old cells, regeneration of all, repairing, regeneration of cells, then for healing, right? So it is helping for repairing of old cells, then regeneration, then healing. All these are the importance of cell division. Clear? So once again, for clarity, I'm saying here this will be quite confusing, this meiosis. So no need to get confused here. I'll tell you, meiosis means there are two steps, meiosis 1 and meiosis 2. In meiosis 1 means this is meiosis 1 is also called as heterotypic division. Okay, because different types of division here you can see, and this is subdivided into prophase 1, metaphase 1, anaphase 1, and telophase 1. We said in prophase 1 this is very long, prophase is very long, and that is very complex. And this prophase is very complex in meiosis 1. So prophase 1 is divided into different stages: liptotin, zygotin, packetin, diplotin, and diakinesis. Okay, so leptotin means diplotin diakinesis. In leptotin you can see long chromosomes. Okay, so chromosomes will show a banquet type of arrangement in leptotin. Then it will move to zygotin. All these things are taking place in a prophase. So it is a long stage and it's a very complex stage. Then the chromosomes from the leptotin or leptonema, will call it as leptotin or leptonema. In that beaded long chromosomes you can see, and the chromosomes will show a bucket arrangement. Okay, it will show a bucket type of arrangement, and then it is moving from leptotin to zygotin. So in zygotin, here you can see the thickened chromosomes in zygotin. Okay, or in which way, like paired homologous chromosomes. Homologous chromosomes which are in paired form, that means bivalent or tetrad forms you can see. And here we said the crossing over, the synapsis, you can see pairing of chromosome that occurs. So bivalent form uh, of the synaptonymal complex to stabilized paired condition, you can see paired condition of chromosomes, in which one, in zygotin or zygonema. Then comes the packeting stage. This packeting stage, in this, among all these, packeting stage is long one, because here the crossing over is taking place, so it is taking time to cross over. So here in the crossing over stage, you can see the tetrads, they will clearly appear as group of 4 homologous chromatids, and here crossing over will get over, and recombination, that and all, in which stage it is happening, in packetin, crossing over and recombination. These are taking place in the packing stage, crossing over and recombination in packetin stage. And this stage is characterized by the appearance of recombination nodule. Recombination nodule in which stage? Packetin stage. Okay. And mutual exchange of the corresponding segments of non-sister chromatids of homologous chromosomes, that is taking place in packetin. See, crossing over, then the non-sister chromatids are getting exchanged. Okay, so recombination, there is recombination, different combinations here you can see in this. See, the whole chromosome we have marked as blue color, and at the end you can see right color. That means the chromosome portions of non-sister chromatids, that exchange of chromosome portions happened due to crossing over. Here you can see the crossing over. So as per that, exchange of chromosome portions happened. Then next comes the diplotin stage. In diplotin stage, this bivalent chromosomes you can see, and here chiasmata, which is the site of crossing over, that also you can see, and points that will strongly appear. And in some, here one important point about diplotin, in oocytes of some vertebrates, this diplotin stage that may last for so many months or years. Diplotin stage, okay, in some in the oocyte of some vertebrates. Then comes diakinesis. This is diakinesis means the terminalization and this terminal chiasma that slips off the end of the chromosome, and this position, it will take the position here. Terminalization occurs. Which one? In diakinesis. So after these long process from prophase 1, it will move to the metaphase 1. The cell will move to the metaphase 1. Right. In metaphase 1, what is happening? Arrangement of chromosome at the equator and maximum condensation of chromosome. Here maximum condensation is happening and chromosomes are arranged at the equator. Right. Then from that it is moving to anaphase 1. Anaphase means you can see the movement of homologous chromosomes towards the opposite poles. You can see here movement of homologous chromosome towards the opposite pole, and chromosome number will become half. Here the chromosome number is becoming half. Right. Next comes the telophase 1, that is the elongation of chromosome formation of two haploid chromosome and the formation of two haploid cells. Here elongation you can see, and the formation of two haploid cells and nuclear envelope that is forming, or the reappearance of nuclear envelope and nucleoli, both are happening in the telophase 1. Okay. Next, these new cells which are produced, that is entering to the meiosis 2. Meiosis 2 is also called as homotypic division. Okay. Homotypic division. Which one? Meiosis 2. And this meiosis 2 is again divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. So all these phases, prophase 2, metaphase 2, anaphase 2, and telophase 2, all these are similar to the mitotic phases, except that these phases occur in two haploid cells. Okay, and this result in the haploid daughter cells. But here you know that recombination is happening, exchange of chromosome portions are happening. So here genetic condition. After mitosis, what you can see, similar two cells with similar genetic condition. But here finally four cells. Here two cells and here two cells are forming, right? So the genetic code, recombination happened. That's why the genetic code differ in all four cells, and that result in the formation of gametes. Formation of gametes, you can see formation of gametes. Four different types of gametes will form, and here then the fertilization of two gametes. When the fertilization of two gametes are taking place, that means male gamete and female gamete is taking place, that completes the new set of chromosome as a result of this crossing over. Okay, and that's how we said the crossing over is happening, and as a result of crossing over, different genetic conditions are forming in each cell, and that makes a source of variation. Okay, different genetic conditions are getting formed in the daughter cells. So here if four daughter cells are forming, means all the four daughter cells will be having different genetic condition, so that makes the variation. Okay, Now we need to learn about the, now it's clear about mitosis and meiosis, right? So we need to know the difference between mitosis and meiosis. Difference between mitosis and meiosis. First difference, in mitosis, gap phase 2, in the interphase, gap phase 2 is normal. In which one? In mitosis. But here the gap phase 2, that is very short, and that is non-existent. That is very short. That cannot exist. Okay, that much short it is. Which one? Gap phase 2. So here the difference, whatever I am saying, each and every point is an important question. This came in the previous year question paper also. Here, the division phase, that is of 1 or 2 hours. Which one? In mitosis. But here in the meiosis division phase, that takes several days to several years. Which one? In meiosis. Clear? And you know that mitosis, that is taking place in somatic cells. Right? But here meiosis, that is taking place in germ cells in the gonads. This is taking place in the germ cells. Okay. So here the mitosis, that is mainly for the body growth, then repairing, regeneration, healing of injured parts, then for the development, embryonic development. At the same time here, this meiosis, that accounts for the formation of gametes in sexual reproduction. Next, one chromosomal duplication. In mitosis, the chromosomal duplication, that is followed by one cell division. One chromosomal duplication is followed by one cell division, and that is producing two daughter cells, right? First chromosome is getting, or DNA synthesis is taking place. That means chromosome duplication. Then it is followed by, after synthesis or doubling of chromosome, cell division is taking place, and as a result of that cell division, two daughter cells are getting produced, right? DNA synthesis, then division, two daughter cells are getting formed. Here, one chromosomal duplication, only one time chromosomal duplication is taking place, and one time DNA synthesis is taking place, right? Or the doubling of chromosome. Then that is followed by two consecutive divisions, two divisions, right? So here four haploid cells are forming. Here two cells, here four haploid cells. Finally, four haploid cells are producing, or they are getting produced. Then mitosis. Here two daughter cells are going to form, and the two daughter cells, both the cells will be having similar genetic condition, or they'll be similar, genetically similar to each other and to the parent cell. Genetic similarity will be there. Right. What about the meiotic division? As a result of meiotic division, daughter cells are getting formed, and daughter cells are genetically different from each other, and it is genetically different from the parent cell too. Okay, some features will be there, not entirely different, I'm saying. Some features will be there, but percentage of difference will be there. Okay, all the four haploid cells which are forming after meiosis, they'll be having similarities and differences. But the percentage of similarities and percentage of differences, that varies. And when it is compared to the parent cell, they're comparing to that also, the similarity percentage and differences, that percentage varies. Okay, so these many differences you can find between the mitosis and meiosis, and it's very important, this mitotic and meiotic stages. So in the next class we'll be discussing about the previous year questions from the cell cycle, mitosis and meiosis. So here main questions which will get, if the cell is in G0 phase, or if a cell is entering into G0 phase, what will happen? I said, while explaining itself, I said that will exit the cell cycle, right? There is no other option. It will exit the cell cycle. It will not move to the division stage or to the next stage. Okay. And what is happening in the post-mitotic gap phase? There, all the other like post-mitotic gap phase means their RNA synthesis, and there won't be any change in the DNA content, but synthesis of histone protein, that will not take place in that. Okay, so here we in the next class we'll discuss one by one, like the topics, mitosis questions, meiosis questions, cell cycle questions, everything we'll discuss in the next class. So here these many topics you need to be clear, especially the stages, meiosis 1 and meiosis 2. And meiosis 1 is again divided into 5 stages: leptotin, zygotin, packaging, diplotin, and diakinesis. So all those things you should know, and which is the reductional division, which is maintaining the same number of chromosome, and what is happening in the meiosis 1, what is happening in meiosis 2, everything you should know very clearly. So if you are learning all those concepts, it will be very easy for you to answer these questions. The picture should be there in your mind, then easily you can answer all these questions. Okay, clear? So we'll see in the next class. Thank you.